Hello and welcome to the Molecular Probe Cell Health webinar series. Today we'll be focusing on autophagy. We will have a question and answer session at the end of the presentation, and we'll address those questions that have been answered during this seminar. Our goal today is to introduce you to the different analytical techniques and the tools that can be used to study autophagy. First, we're going to describe autophagy and the role it plays in cellular function. Then we can identify key features of the autophagic pathway to determine which steps are open to interrogation and what tools we might use to gain insight into the processes. These tools include a range of organic dyes and fluorescent proteins used to analyze live cells in real time or fixed cell preparations. The analysis steps will include quantitative methods and multiplexing to correlate autophagy with other cellular functions that may be of interest to researchers in this area. As we detail each approach, we'll provide tips and tricks for achieving the best results. The name autophagy, which literally means self-eating, reflects its original identification in the 1970s as a survival process activated under conditions of nutrient deprivation. A more current understanding of autophagy includes its role in cellular homeostasis, as well as in organism development and protein quality control. Autophagy has also been shown to be important in defending cells against invading pathogens, and failures in the autophagic pathway can lead to cellular damage and disease. While autophagy is critical in clearing aging or superfluous proteins, aggregated proteins, and invading pathogens to enable the healthy functioning of cells, autophagy is also involved in mediating cell death, as demonstrated by examples where autophagy has acted as a bona fide cell death mechanism. Autophagy degrades large cargo, for example damaged organelles or pathogens, and is also the primary mechanism that a cell employs to degrade long-lived proteins. Many signaling cascades regulate autophagy. Commonly these involve mTOR and AMP kinase. Early in the process, double membrane structures are formed which become the autophagosome. These are known as the isolation membrane and believed to form from areas of contact between the mitochondria and the endoplasmic reticulum. The isolation membrane formation requires a number of well-characterized molecules, including one of the early autophagy molecules, ULK1 complex. This contains both ULK1 as well as a protein named FIP200 and some of the ATG proteins. Many of the proteins involved in autophagy are known as ATG, reflecting their role in autophagy. As well as the ULK complex, also a class 3 PI3 kinase complex containing the VPS 34 and 15 is required. This complex also contains Beckley1 and ATG14. There are a number of other molecules such as ATG9, as well as the PI3 binding proteins WIPI1 and Z51. The LC3 protein is involved in the formation of the autophagosome. Recruitment of LC3 to the nascent autophagosome involves cleavage at the C-terminus by the cysteine protease ATG4. LC3 is then conjugated to PE by a series of steps involving the proteins ATG7, as well as the ATG5-12-16 complex. Whilst LC3 is the most commonly studied molecule, and indeed the isoform LC3b is the one that we'll focus on today, there are other isoforms, LC3a, C, as well as homologs GATE16 and GABARAP. LC3 in yeast is known as ATG8. Later on in the process, you can get recruitment of various binding proteins, one of which is P62. P62 is involved in recruiting polyubiquitinated cargo into the autophagosome and was one of the first autophagy receptors to be identified. The autophagosome itself then forms. This occurs through elongation and extension of the isolation membrane and subsequent fusion to form the characteristic double membrane structure that is a hallmark of a formed and fully mature autophagosome. Following autophagosome formation, the lysosome fuses with the autophagosome, and this brings in the degradative cargo, the enzymes that allow the cargo within the autophagosome to be degraded. The fusion of the lysosome with the autophagosome involves the snare proteins VAMP8 as well as syntaxin-17 and also requires a cytosolic bridging factor, SNAP29. Once the lysosome is fused with the autophagosome, LC3 will be cleaved from the cytoplasmic face of the autolysosome, again by the cysteine protease ATG4. Once all of this has occurred, cargo degradation occurs by degradation by enzymes that are present in the lysosome. So now we've gone over a few of the basics processes involved in autophagy and the role of autophagy in cellular health and disease. We can look at some of the tools that can be used to measure autophagy. These include antibodies against proteins such as LC3, fluorescent protein chimeras of LC3, as well as tandem reporters that tell us about autophagosomal maturation. One can also look at P62 recruitment through the use of fluorescent protein chimeras of P62. And of course, given the important role of lysosomes in executing full autophagic flux, markers of lysosomes are also important, and these can constitute antibodies against lysosomal resident proteins, as well as fluorescent proteins targeted to the lysosome, and indeed various dyes such as lysometropic compounds like lysodraca, which selectively accumulate within lysosomes. Before we go on, there are a few practical considerations to take into account when performing autophagy research. Generally speaking, if you're using a marker such as LC3 to look at autophagosome formation, at rest you have a low autophagosomal intensity. You'll see very low levels of LC3 staining. You'll often, during the course of an autophagy experiment, see an increase in the autophagosomal intensity. This can have one of two causes. One can be the induction autophagy, but it can also arise from an inhibition of autophagic flux. And this is a useful paradigm to be able to tease apart during your experiments. The way in which one can do this is to use inhibitors of lysosomal function. Here on the left we can see that prior to treatment, the cell has few autophagosomes reflecting a low LC3 staining intensity. Upon treatment, the observed result will be an increase in LC3 staining. To dissect whether this is due to true induction of autophagy or inhibition of autophagic flux, one can do this experimental treatment in the presence or an absence of an a lysosomal modulator. If the treatment in question, either drug addition or indeed siRNA knockdown, is inhibiting autophagic flux, one will see no increase in the autophagosomal labeling in the presence of a lysosomal inhibition compared to in the absence of lysosomal inhibition. If the experimental treatment is actually stimulating autophagic flux, you'll see an increased autophagosomal labeling in the presence of a lysosomal inhibitor versus when you don't have that lysosomal inhibitor present. And you can see from the cartoon schematic representation of this below. You can see the increase in LC3 staining in those cells under conditions which truly stimulate autophagic flux. As we mentioned in the introduction to techniques to measure autophagy, one key way of looking at autophagy is with antibodies against LC3 molecules. And again, LC3b is one of the most commonly used markers. We're going to primarily focus on imaging applications, although you can use antibodies against LC3 in microplate assays or indeed Western blotting. More details of our microplate assay can be found online on our website. And this uses a time-resolved FRET interaction between GFP-LC3 and a lanthanide-labeled anti-LC3 antibody. LC3 antibodies can also be used in Western blot applications, where one looks at the change in mobility shift of the LC3 protein following cleavage and conjugation with phosphatidyl ethanolamine. When we look at imaging of LC3, you'll see these data from the above slides. Essentially, we use, in this experiment, chloroquine to block autophagic flux. And at low doses of chloroquine, we see very low levels of LC3 staining on the left. However, if we use higher concentrations, around about 60 micromolar of chloroquine, we see a robust accumulation of LC3 positive puncta within the cytoplasm. This is primarily a qualitative measure of autophagic activity, and it's often important to quantify this. There are two ways in which one can quantify autophagy. You can perform spot counting experiments. These can be either done manually or using automated analysis algorithms. A quantification of autophagy can also be taken just by looking at the intensity of staining, and this is predominantly done by looking at intensity in a perinuclear region. And here we just have two examples, one where we've counted the number of autophagosomes on the left, and on the right just looking at the intensity of staining under identical drug treatment conditions. and both give a nice quantitative measure of the degree in which autophagy is occurring. Because of the role in autophagy in regulating a number of different cellular functions, it's often important to multiplex a measure of autophagy with another cellular process. Antibodies are useful against LC3 in this context because you can use antibodies against other cellular markers and have a look at a more complete picture of the processes occurring within the cell. These can include markers for proliferation, viability and apoptosis, as well as looking at sequestered cargo such as pathogens or protein aggregates. The data on the right here shows one of our metabolic labels for protein lifetime, Clickit homopropradial pyromycin. We'll show a little more data on that in the subsequent slide. Here we've labeled cells with Herbst 33342 to label the nuclei, and we've also fed them homopropradial glycine. This is incorporated into proteins as they're synthesized, and then if one chases out in HPG3 media, you can start to look at the intensity of HPG signal as a function of protein lifetime. Here, in the presence of an inhibitor of autophagy, we see a dose-dependent accumulation of protein within the cell, reflecting the inhibition of protein clearance by this inhibitor of autophagy. We can also co-localize these protein aggregates that form within cells with markers of the autophagosome. Here we've used an Alexaflor 647 secondary antibody to label autophagosomes, which are marked by LC3b antibody. We can also look at other cellular processes such as proliferation. This is particularly important in processes such as cancer, where the cells will undergo uncontrolled proliferation, and indeed autophagy has been implicated in the progression of cancer in a number of models. Here we've used our click marker of proliferation, Clickit EDU. EDU is incorporated into cells that are undergoing proliferation, and by looking at these along with measures as well as markers of autophagy, we can start to look at to what extent autophagy is occurring or not occurring in these highly proliferative cells. Autophagy is a highly transient process. The half-time of an autophagosome is around about 25 minutes, and so it's often important to do these in live cells and do time-lapse imaging. One mechanism by which you can look at autophagy in live cells is to use LC3 fused to either GFP or RFP. It's important when doing these experiments to consider that you're often overexpressing the protein of interest, and for this reason we include a control. It's a non-cleavable version of LC3. This allows to control for expression artifacts. Essentially, during the processing of LC3, it's cleaved at glycine 120 by ATG4, and this allows the conjugation of LC3 with phosphatidyl ethanolamine. If the glycine at 120 is mutated to an alanine, the cleavage cannot occur, and so LC3 cannot be recruited to autophagosomes. As I say, this serves as an important control for expression levels, as LC3b itself will tend to aggregate upon overexpression. Here's an example of time-lapse imaging of autophagy in U2S cells. Here they've been nutrient deprived and their cells are expressing the RFP version of LC3. And one can see over time the formation of an autophagosome. Here we've acquired images every 2 minutes to fully capture the process. If we look at the region indicated by the white arrow, this is the site where the autophagosome forms. And we can see it forming over time and moving throughout the cytoplasm of this cell. Much like with antibodies, we can also multiplex when using fluorescent proteins. Here we can use a marker of the microtubule cytoskeleton, a tubulin GFP, and look at the co-localization of RFP-LC3 with microtubules. This is of course important when looking at the trafficking of autophagosomes around the cell and indeed their subsequent fusion with the lysosome. We can also start to look at the actual active processes in which autophagy is carrying out. Here, this is a specialized form of autophagy known as mitophagy, whereby damaged mitochondria are engulfed by autophagosomes and lead to their clearance. This buffers the cell from aberrant ROS production by damaged mitochondria, as well as helps to restore the energy balance of the cell. By using a GFP targeted to the mitochondria, in conjunction with RFP-LC3, we can depolarize mitochondria with a low concentration of a protonophore. We see these characteristic mitochondrial spheroids produce these are very small rounding up mitochondria, And indeed, these are the mitochondria that are co-localized with the marker of the autophagosome, RFP LC3B. We can multiplex with other processes as well, such as apoptosis. Here we've used our cleavable substrate that detects activation of caspase 3-7. And indeed, we can look at the concurrent activation of autophagy and apoptosis after treatment with a drug, staurosporin, which induces apoptosis. Using this selection guide that's available on Life Technologies' website, you can compare the properties of many of the tools that can be used for LC3B detection and tracking. The primo autophagy sensors can be expressed and allow imaging in live cells of autophagy. They're available in either green or red fluorescent versions. The antibody can be detected with a wide choice of secondary labels, giving you a little more freedom in terms of what colors to detect the autophagosome by, although it does require the cells be fixed and permeabilized before application. In the web tool, you'll also find details of our TR-FRET-based plate reader assay for high-throughput screening. Another key process in autophagy is the maturation of the autophagosome to the autolysosome. We've developed a tandem sensor that allows that maturation to be visualized in live cells over time. This sensor works on the basis of a differential pH between the autophagosome and the autolysosome and the lysosome. The autophagosome forms with alumina the pH around about 7. The lysosome itself has a pH of around about pH 4. As the autophagosome fuses with the lysosome, the pH of the LC3B positive structure drops to that of the lysosome, so around about pH 4. By using two different fluorescent proteins which have different acid sensitivity, both of which are attached to LC3, you can track the maturation of the autophagosome by the loss of the pH-sensitive GFP, whereas the RFP, which is less pH-sensitive, the fluorescence from that is retained throughout the pathway. And so the neutral autophagosome has fluorescence from both RFP and GFP, whereas the acidic lysosome has fluorescence only from the RFP. What this looks like in real cells, you can look at the number of RFP positive, the number of GFP positive cells. Vehicle treatment, there's no accumulation of LC3, so you see very low numbers of both RFP and GFP positive cells, although you do occasionally see these spots, and these are often just reflecting basal autophagy. If one uses chloroquine to inhibit autophagic flux and accumulate LC3B positive puncta within cells, you see both RFP and GFP positive cells. This is because chloroquine acts by neutralizing lysosomal pH. This means the lysosomes that fuse are not acidic, and so you don't get a quenching of the GFP fluorescence. If one blocks autophagic flux, thereby accumulating LC3B positive puncta via a mechanism that inhibits lysosomal enzymes, this can be done using drugs such as lupeptin A, the pH in the lysosome is still highly acidic, and so you'll get a loss of GFP positive spots, but a maintenance of the RFP positive spots. And this also allows live time-lapse imaging of these processes. If one serum stars using Earl's buffered salt solution, we can see the rapid formation of both green and red positive structures within the cells, which over a period of time lose their green fluorescence and retain only their red fluorescent protein. So this is the maturation of the autophagosome to the autolysosome imaged in live cells. If we refer back to our selection guide, you can see that the Tandem sensor can be used to monitor autophagosomal maturation in live cells. Imaging will require the presence of both a TRITSI and a FITSI filter, so these are orange and green filters on your microscope. And of course, the protein can be fixed for subsequent multiplexing, although one needs to be careful that the pH environment around the proteins hasn't been changed. P62 is one of the best characterized receptors for specific protein degradation. It is often used both as a marker for ubiquitinated cargo, as well as a general marker for autophagic flux, since it is continually cleared via macroautophagy. As we mentioned previously, P62 binds polyubiquitinated cargo and delivers it into the autophagic pathway by binding to LC3. Indeed, under conditions of blocked autophagic flux, one sees a co-localization between P62 and markers of the autophagosome such as LC3. Here we show cells expressing GFP-P62 as well as RFP-P62 that have been treated with chloroquine and fixed prior to staining with Alexa fluor-conjugated anti-LC3B antibodies. Co-localization of both P62 and LC3B confirms that these aggregates are found within autophagosomes. And here's some quantification of cells expressing either a GFP-P62 or an RFP-P62 using chloroquine to block autophagic flux. We see a significant increase in the number of P62 positive puncta per cell. And because we're using RFP or GFP chimeras to P62, we can look at the accumulation of these structures over time in live cells. Here we have a cell expressing GFP-P62, and we've induced autophagy with serum starvation in the presence of an autophagic blocker, chloroquine. We can see the time-dependent accumulation of P62 positive puncta within the cells. You'll see them accumulating within the cytoplasm. Again, if we use our selection guide to look at the various properties of these RFP and GFP chimeras, they're primarily used in live cells, although they can be fixed and multiplexed with other antibodies. Lysosomes are critically important for autophagy. They contain the hydrolytic enzymes that break the cargo down, but it can also serve as signaling and sensing platforms. Molecules such as mTOR are present on the lysosomal surface. Of course, mTOR is a key signaling molecule within the autophagic pathway. Strategies for visualizing and tracking lysosomes include specific antibodies, organic dyes, and targeted fluorescent proteins. Antibodies tend to be against resident membrane proteins such as LAMP1 or LAMP2, as well as LIMP. These are lysosomal integral membrane proteins, and the LAMP stand for lysosome-associated membrane proteins. One can also have antibodies against specific enzymes within the lysosome. A commonly used antibody here is an antibody against cathepsin. Various organic dyes can be used to label lysosomes. Some of the more commonly used ones are lysotracker dyes. These are lysometropic compounds that accumulate in acidic structures, and we have them available in green, red, and deep red versions. So those are fluorophores that fit in the FTSE, TRITSE, and STI5 type channels, and are very useful for multiplex analysis in living cells. Fluorescent proteins can also be used to target lysosomal membranes, and these commonly are GFP or RFP chimeras against LAMPs or LIMPs. Here we show an example of multiplex lysosomal structure and function. Here we use our lysosome GFP as well as the lysotracker dye, lysotracker deep red. We can see a very nice labeling of the lysosomal membrane with lysosome GFP, and we see that lysotracker deep red is lodged right in the lumen of the lysosome, confirming the specific labeling of the lysosome with lysotracker deep red. Of course, one can use these labels to multiplex in the context of autophagy. Here we have the autophagy sensor LC3B GFP in conjunction with an RFP targeted the lysosomes. And using these two approaches, you can watch the fusion of the autophagosome with the lysosome. You can also use the lysotrackers to visualize the same process. If we look at the Tandem sensor, so this is an RFP and GFP LC3B that we described before. It can be used with a spectrally distinct lysotracker deep red. In control cells, we see a large number of blue spots. We've pseudo-colored the lysotracker label blue, and we see a few yellow spots reflecting autophagosomes and a few pink spots which show the co-localization between lysotracker deep red and the acid-insensitive RFP of LC3B, RFP, GFP. If we nutrient deprive, we do serum starvation in Earl's buffered salt solution. We see the rapid formation of yellow puncta, which then subsequently become pink as the blue lysotracker fuses with these structures. We see a loss of the green fluorescent protein. We can also just conclude by looking at some of the selection guides for the lysosomes. Here we have the properties of the lysosome GFP and RFP. Again, these can be used to multiplex as well as label live cells. And of course, they can be fixed if one wants to combine with an antibody. The Lysotracker compounds are available in green, red, and deep red. These are used to label live cells. They're not well retained following fixation, and so they're not ideal to use with antibodies, but they are very nice to multiplex with other labels in living cells. For more information about autophagy, you can go to our webpage at lifetechnologies.com/autophagy, as well as contact our technical support groups who can give you more information. There are also references available to download at this site if you're interested in further reading about autophagy. Now, following the webinar, we'll attempt to answer some of the questions that may have been posed. I believe we just have the one question come up. The question asks, can these antibodies be used on ex vivo purified human samples? And is it sensitive enough for cells not undergoing starvation? So the antibody that we have does recognize human LC3, and so if you have purified protein from human samples, it will recognize it, as well as cells or tissue from humans. As I say, the antibody does show species reactivity with the human LC3. The sensitivity, it's going to depend very much on the extent to which autophagy is occurring in these samples. But the antibody itself is relatively sensitive to pick up low levels of autophagy that might be occurring under basal autophagy. I'm just going to have a look to see if any more questions have come in. We have one question asking about macrophages. Macrophages are particularly difficult. It's very difficult to deliver genes to this cell type, and so our fluorescent protein-based sensors are not ideal here. Probes such as Lysotracker work very nicely in macrophages, and indeed one can fix these and use antibodies against LC3 along with antibodies against lysosomal markers to look at the presence or absence of autophagosomes and autolysosomes. We'll stay online for several minutes afterwards if there are any more other questions to come in. But I'd like to thank you for tuning into our webinar today, and we hope you found it useful. Will this webinar be archived for future reference? Just came up. I believe that it will be. Most of the webinars that we've delivered in this series are available on our website. We have a number of webinars looking at tools for viability, as well as apoptosis research, and then other webinars that looked at more cell structural processes within cells, cytoskeletal function, and also a basic webinar on tips and tricks for immunofluorescence. And all of these are available for download at our website, lifetechnologies.com. Okay, we have another question come in that's pertaining to using antibodies in combination with FACS. FACS analysis is a little more difficult when looking at autophagy because most of the techniques we've described deal with not so much changes in intensity, but changes in localization. There have been several ways around this described. Essentially, if one does a mild permeabilization prior to fixation, typically these use detergents such as saponin or digitonin. When LC3 isn't associated with the autophagosome, it can be thought of as being soluble, and so this mild permeabilization washes out the cytoplasmic component. If autophagy has been upregulated or indeed autophagic block occurred, you'll have LC3 associated with the autophagosome. And this is insoluble to the saponin or digitonin treatment, and so you'll see increased binding, and that way you sort of falsely impose a fluorogenic response using the antibodies. So to use in combination with flow cytometry, one wants to try and wash out the cytoplasmic component. And there's several papers published, one in particular in the journal Autophagy, details the exact protocol for doing this. We also have another question about the marker for mitophagy. We actually recently published a review on tools and techniques for looking at mitophagy. It will be available in the November issue of Autophagy. Essentially, here you want to label both components of the autophagic pathway, LC3 and P62 are particularly relevant here, as well as mitochondria. Antibodies against mitochondrial proteins are very useful in this context, as well as probes such as our mitotracker reagents. These are organic dyes that specifically label mitochondria. And of course, mitochondrial targeted fluorescent proteins are useful. You can then also combine a mitochondrial marker with markers of autophagy, such as LC3, with lysosomal markers to confirm that your damaged mitochondria is being trafficked to the lysosome. These are all useful probes to look at when investigating mitophagy. Okay, well, I think that concludes our question and answer session. I'd like to thank you again for tuning in for this webinar, and we look forward to seeing you for subsequent webinars. Thank you very much. Bye-bye.